The cell cycle of a skin cell, written by me. The first phase of the cell cycle in a skin cell is gap 1 as part of interphase. It usually always happens as skin cells continue growing in us, but is especially visible when one cuts open their skin to reveal a wound. The cell sends signals to the niche cells to produce more recovering skin cells. Anyways, let's talk about interphase. Interphase is the first of 5 phases of mitosis, the division of the nucleus. In interphase, there are 3 further divisions. There is G1, S, and G2. G1 is the most time-consuming phase of all of interphase. During G1, the mother cell is pretty much as original, but it begins to grow in size and produce more and more and more and more organelles. They develop an extra set of chromosomes and will continue storing them until the next phase, which is synthesis. Synthesis is the second phase of interphase and the second phase in preparation for mitosis. It is where the cell begins to reproduce its own DNA and 2 copies of each microtubule are created. The synthesis phase starts when DNA replication begins, and when that is done, chromosomes replication starts. The chromosomes create 2 sister chromatids that are attached at the center. By this point, the DNA replication process has finished, and that is when we move on to the last phase of interphase, also known as gap 2. The third and last phase of interphase is gap 2. I know, interphase has felt really long, but that is because interphase takes up about 90% of the time of mitosis. In this phase, DNA and chromosomes have all pretty much finished synthesizing. The main event happening in this phase are further preparations for mitosis. The cell continues to synthesize some protein and continues to grow in size. This wraps it up for interphase of cell division. We can finally get started with mitosis. Interphase was pretty long, so let's try to keep it short for mitosis. Mitosis is where nuclear division happens. The cell has reached its maximum size, so let's start dividing away. The first phase of mitosis is prophase. In prophase, the chromosomes condense and become shorter and thicker, forming sister chromatids. They begin migrating to the center of the cell. The nuclear envelopes break down as the microtubules move to opposite ends of the cell, forming spindle fibers. Now, onto metaphase. In metaphase, the nuclear membrane disappears completely. The spindles complete their development and connect the chromatids to the microtubules. The sister chromatids all line up at the metaphase plate. The metaphase plate is the line that is an equal distance from both poles of the cell. Now, onto anaphase. The third phase of mitosis is anaphase. In anaphase, each individual sister chromatid separates at the centromere and go to opposite sides of the cell, or opposite poles. The spindle fibers that are not connected to any chromatid help the cell elongate in preparation for cytokinesis, but that happens further in the next phase, telophase. Telophase is the final stage of the nuclear division, aka mitosis. The chromosomes reach opposite ends of the cell and begin to uncoil in 2 separate nuclei. By then, the spindles have completely disappeared. Cytokinesis begins midway through telophase, which leads me to the last phase, cytokinesis. Cytokinesis is the last step of the cell division process, and it is where the cytoplasm divides, hence the name. The result of this is the formation of 2 identical mother and daughter twin cells.